Hey guys, welcome back. So, second day, surviving the digital schooling, I hope. I haven't been flooded with emails from you guys, so I assume that you must be getting along okay. Last I checked, 20 people had finished the Edpuzzle video all the way through, so awesome job there. If you guys do have any questions, my email is rstecker@rcchristian.org. Okay, so take a minute, pause the video, write that down. You can shoot me an email anytime you need if you're stuck on something or whatever. And I check it throughout the day and on into the evening, so I'll get back to you pretty quickly. You can also send me an Edmodo message directly, or even just post it to the bio page. I'll help you. Someone else will help you. However, and we'll get it sorted out just fine. So, at any rate, today what I'm going to do is I'm going to take you guys through real quick and in a hurry how to make your lab report. We had talked last week that we were looking at probably Monday your plates would be done. Friday would be our due date for the lab report. And there's really not a reason we can't stay on that same schedule. So we're going to stick on that same schedule. And so today what you all got in your email was a picture that looked like this, okay? Now, your picture probably said your name at the top. If you didn't get this picture, go to your rcchristian.org email and look for this picture. Every single student who had a plate in the lab, even those that weren't in the incubator and were laying around in random places, even those kids got their picture sent to them. And so go to your rcchristian.org email. This is a spare. I'll put the spare image on Edmodo. So if for some reason your plate got lost, you can use the spare image. I actually have two spare images, and hopefully your computer or phone has the capacity to zoom in. If it does, your life's going to be a lot easier. If it doesn't, then it's not going to be that easy, but it's still doable. So these are going to be your results for the lab report, and today I'm going to take you how to do everything except for build the graph. So let's jump right into it. How to make a lab report, assuming you haven't started. And I know that at least one of you is over halfway done, and I've helped a lot of you start. But let's say you haven't even started. Let's start a lab report. Here we go. First things first, go to the center and think of an appropriate title for your lab report that's reasonably descriptive. A bad title would be lab report by student. Okay, put your first and last name, right? But don't call it lab report. That's a terrible title, right? Let's call it comparing bacterial biodiversity in Rapid City Christian School? At Rapid City Christian School? I don't know. Let's pick one. There, I like that one better. Comparing bacterial biodiversity at Rapid City Christian School. Or for my sample, I could pick what part of the human body has the most diverse bacteria, because as you saw in my sample, it was like foot and arm and that kind of stuff. So once you have your title, jump back over and go to your left margin, and let's throw a couple bold headers in. Let's go abstract. Let's go introduction. Let's go materials and methods. Let's go results. And let's go discussion and conclusion. Now, this is your first time going through this stuff with me, so if it seems like I'm repeating things, don't panic. I know that for students, lab reports can be a big hurdle. So abstract, your teacher said, save that for later. So we're going to save that for later. So let's jump to our introduction. Introduction, we're going to jump down here. We're going to take our bold away, and we're going to go back up and tab in here. So one space down, tab over, introduction. We're looking for things like a hypothesis. We're looking for things like relevant background information, okay? We're looking for things like why you're doing the experiment. Okay, so go ahead and write that down if you haven't. Here's what this could look like. Okay, pause the video, write those down if you like. Let's go ahead and write a quick introduction, shall we? We're going to stick with some common knowledge stuff because I don't have any APA citations rolling around in my head right now. How about this? Bacteria are single-celled organisms that live essentially everywhere. Okay? Relevant background information. This experiment was conducted to Okay, now bear in mind, I can't say I conducted this experiment. This experiment was conducted to finger, arm, mouth, foot. Okay, so let's say was conducted to test which part of the human body has the most bacteria. Maybe we should even say the most diverse bacteria, but that's fine. So we have to make a hypothesis. I didn't make a hypothesis because I didn't plan on writing a lab report. You were going to write the lab report, but here we sit, and I'm writing the lab report. So let's do this. Foot, mouth, finger, arm. I would probably say mouth. Now, I can't say I think or I hypothesize. Let's say it was hypothesized that the mouth would have the most diverse bacteria, pardon me. Okay, so we have our introduction, what we do, what are we doing, why do we do it, and a hypothesis. Okay, your introduction could be longer than that, but let's just jump on, shall we? Materials and methods, moving on, materials and methods. Okay, past tense narrative, exactly what you did. What did you do? How did you do it? Step by step, as a past tense narrative, not as an instruction manual for how to put together Legos, okay? We're talking about a past tense narrative. So let's say agar plates were made from beef agar according to manufacturer's specifications. How about that? We probably wouldn't need to say that on like a college level lab report, but for a high school kid who's going to conduct your same experiment, we should probably throw that on there. Plates were allowed to sit for 24 hours, and the agar cooled and set up. How's that sound? Set up as a solid. Okay, now bear in mind, I'm trying to show you this as a model. Don't copy my sentences word for word, right? Don't do that. Make your own sentences that tell me what you did and how you did it. Let's say Q-tips were used. Q-tips is a trademark name. I should say cotton swabs, shouldn't I? Whatever. We'll plagiarize their copyrighted name. Why not? Q-tips were used to swab bacteria from an arm, a finger, a mouth, and what's my last one? I can't see. Oh, and a foot from the same person. And bacteria were transferred to the agar plate. Okay, so now you're going to go on, and you're going to say that the agar was allowed to incubate for 5 days, or whatever the date I had you guys write down in your lab report was, at a certain temperature. Our temperature was 22 degrees Fahrenheit. Really should be in Celsius because everything's done with Celsius in science, but that's, oh, hold on. It is Celsius. Oh my goodness. I'm sorry. I'm sorry. Whoops. There's a downside to making the videos this late at night, right? Of course it's 22 degrees Celsius. If you incubated bacteria at 22 degrees Fahrenheit, it would be a frozen brick. Oh my goodness. Okay. So anyways, go on to describe exactly what you did and exactly how you did it step by step. Avoid words like I, me, Mr. Stecker, made. Don't do that kind of stuff, okay? So we go through our materials and methods. Now, results. Here we go. Results. Bear in mind, you guys are doing this on a photograph. I'm not going to go to the school, count your colonies, and say, ooh, your results said 28 colonies, but there were 29. So you're getting docked for that. No, you're not. But, let's use our judgment here. It looks like the foot had some white colonies that grew. So let's just run a quick count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30. Okay, should we say 35? I'm thinking 35. Okay, some of them have kind of grown into each other. It's hard to get a good count. Let's say 35. Okay, so results are a boring section, but you're going to literally just give the data. Don't say what it means. We're going to make a graph tomorrow because that alone in Excel is a project all of its own. So let's say this. The foot sample produced, what did we say, 38? I feel like we said 38. I don't know. 38. All right, now jump to Edmodo and you'll see this diagram. This is not an Edmodo post. This is very helpful. Now, from this picture, I'm saying circles. We could be wrong. If we zoomed in further, maybe I'm wrong. But I feel really confident about circles. So we're going to say 38, circular. Now, you might not get a raised or a convex from the photograph because it is going to be essentially a 2D photograph. So let's just go with what we know and not guess at what we don't know, right? This is science. So we're just going to say circular. 38 circular yellow colonies. That's it, okay? That's all you have to do. Now, the next one, we're going to jump over here to the finger sample, okay? Zoom in again, and you can count that guy's 1, that's 2, 3, 4, 5. Now, these are kind of grown together. So it might even be worth, maybe we'll specify this and say 2 small colonies and 2 very large colonies, right? And to kind of let people know what's going on. And this photo can go in results too. So let's go ahead and do that. Whoops, that's bolded. I don't want that bolded. I want that to be, here we go. What is this sample? Oh, finger. Okay, sure. The finger sample produced 2 small white circular colonies and 2 very large white colonies. And here's the part where it's going to be kind of tricky because it's kind of up to you. Do you think these were all circles that grew together, or do you think that they have a margin that should be something like undulate? You know, that's a judgment call on your part. If you want to say they're circles that grew together or that it's an undulating margin across the whole thing, I'm not going to dock you for that, right? The point is that we're learning to make a lab report. So let's say it's 2 undulating colonies. Why not? With 2 very large white colonies with undulating, what would you call it? Borders? Forms. Sure. Okay, perfect. And then you're just going to keep going. So let's jump over here, and you're just going to keep going around your plate. And if you've got, this guy's a good one because it's got some yellow, it's got some white, it actually has an orange, and it has a red. And so I'm going to say 1 orange colony, 1 red colony, 14 yellow colonies, and 1 white fuzzy mold. You know, I'm literally going to lay that out. Just here's the results. It's that straightforward. And I will do that later when I show you how to make the graph. And we'll be in great shape, right? So discussion and conclusion is going to be a little bit tough for us to do right now because we don't really have our results. We could actually look at these results and figure it out. I'll show you how to make a graph next time. Oddly enough, the mouth was super clean. I don't know. I think it's because I drink too much coffee and just that acid kills bacteria, I suppose. But who knows, whatever. But honestly, just looking at this, yellow, white, white, yellow, red, orange, I feel comfortable actually helping you write a discussion and conclusion right now. So let's just jump down here. We're going to have a graph, okay, to be continued. Oh wait, you have to have a dot, dot, dot. Ellipses, I'm told they're called. Hopefully James is right. Who knows? He's not going to make me look dumb. I already said we incubated bacteria at a freezing 22 degrees Fahrenheit, right? Too late for that. That ship has sailed. Okay, so let's do a quick discussion and conclusion, right? You finally get to say what your data actually means. And so since we have an arm that's obviously diverse, this should be super easy. So we can go ahead and do this. So let's see, how should we do this? Our original hypothesis was that the mouth would be dirty, right? Because mouths are dirty places, I would assume. But apparently, maybe not as well. So let's do a quick discussion and conclusion. So what did our results show? Past tense, here we go. The results showed, should we do a little surprisingly, there we go, we're kind of letting them know our hypothesis was proven wrong. The results showed surprisingly that the mouth grew no colonies at all. The arm, was that correct? Yeah, the arm, however, grew howevery? Goodness gracious. Okay, the arm, however, another comma, grew, what should we say, 4 different types of bacteria. I like that. And was the most diverse. Wow, typing in real time is just is a struggle apparently for me. I apologize. All right, so the results showed surprisingly that the mouth grew no colonies at all. The arm, however, grew 4 different types of bacteria and was the most diverse. So that gives our basically our discussion and conclusion. We could say that the finger and foot, if you wanted to try to explain them, each grew 1 type of colony, but that's already really laid out in your results. So I don't know if we really need to go back and say that. So the results showed surprisingly the mouth grew no colonies at all. The arm, however, grew 4 different types of bacteria and was the most diverse. So now comes the hard part. We have to try to explain our data, right? So let's explain this. We can zoom in a little bit so we can see closer here. You can see what I'm writing. There, that's better. All right. Oh boy, you might use the word the author or the authors in the discussion and conclusion. I think we can avoid that by saying this could be caused by, boy, I don't know, how would we explain that your arm has more bacteria than your mouth? I don't even know how to explain that. We can't go with the surface area because your mouth has a huge surface area. Arms aren't more humid than mouths. So let's just go with the funny thing I threw out. Like, why not? This could be caused by excessive coffee intake from the participant. This highly acidic environment could potentially slow the growth of bacteria. Like, why not? We're running with the only stupid idea I threw out was that I drink way too much coffee, and we're gonna run with it. Let's do it. It's our lab report. We can do that. So the other thing we want to do, we want to bring it back to our hypothesis, which I feel like we kind of did. Did we state that? Okay, so let's say the hypothesis, the original hypothesis, that the mouth would be the dirtiest. I don't really like to say dirtiest. It should be the most biodiverse, but whatever. Was, should we say, proven incorrect, refuted? Let's say not supported. We don't want to say proven. No, we don't prove. We provide evidence for. This is science, not the Mythbusters. Come on. All right, so the original hypothesis that the mouth would be the dirtiest was not supported by these results. So now we got to do the embarrassing part and talk about experimental errors, what future scientists should examine, that kind of stuff, suggestions for future research. And so there's a lot of different ways we could take that. I'll probably let you guys customize that to what you want to do. There's lots of glaring mistakes that I'm sure we could address. For example, our Q-tips weren't sterilized Q-tips. They were just your plain old cotton swabs from the grocery store. There's a lot of different ways we could tackle that, right? Future scientists should study, whatever, the arms of different people. Why not? Like, let's study many arms. There's lots of easy suggestions you can do to make people aware of what future researchers should do. So we'll just throw a couple notes in here for your guys' sake, okay? Future researchers, what should they be doing? Experimental errors, what went wrong, those kinds of things. Go ahead and put that in for what we can do for a discussion and conclusion. And like I said, tomorrow I'll show you the graph, but for right now, let's just stop right there. So what I'd like you to do, really there's no lecture notes for today on viruses or bacteria or any of that. Go ahead and write this. Leave the abstract alone. Write your own introduction. Don't copy mine. Write your materials and methods. Write your discussion and conclusion. And tomorrow we'll come back and we'll tackle our graph. And then we'll bring it all back in for a landing on the abstract. Probably the toughest piece of this to write because you do have to be so succinct. But we can do it. We'll just take one little baby step at a time. If you have any questions, let me know. I will put photos of samples. If you didn't get an email or something went wrong, use a spare. Okay, you can do that. And this other image right here is already on Edmodo. I can put that in the same post for Tuesday's lesson plans. I want one Edmodo post per lesson plan. That way you guys cannot be lost, okay? Unless, the other 60 million kids using Edmodo this week causes Edmodo to slow down or crash, in which case we'll just limp along as best we can, okay? There's nothing magical about Friday. If you need to extend this into the weekend to finish your lab report and do the best job you can, by all means, I'm going to be flexible and work with you. But don't binge watch every episode of every show on Netflix and come to school with nothing done and say, I couldn't do it because the coronavirus crippled a nation. You're going to be fine. Just take it one little baby step at a time, and you guys are going to do great, okay? So I'll see you in a week. And if you have any questions, drop me an email, okay? Thanks a lot. Talk to you later. Bye.